Hi, this is Phil Newman from Longevity.Technology, and I'm delighted to say that I'm joined by Jamie Hayward, who's the founder and CEO of Alden Scientific. So, Jamie, hey.
It's great to be here.
Well, Jamie, delighted to have you here, and I know that you've done a lot of work in the biotech scene before. This isn't your first rodeo, but I'm keen to understand, first of all, why you started Alden Scientific, and secondly, kind of what is Alden Scientific. So perhaps you can help me out with that.
Yeah. So I've been involved in discovery and development for about 25 years. It started when my brother had ALS, and I founded a research institute. And, I worked very hard to cure his ALS, which is what ALS TDI was founded to do. And, we did the first stem cell transplant in the field 23 years ago. And some of our work was involved in the start of the replication crisis because we showed that there were issues with mouse studies and replicability that really transformed the way certainly neuroscience, animal research, and I think largely animal research was done. And the theme that came out of that was that I think an underestimated problem in any biological problem is knowing what to do and whether it's working. And, when I was facing this with my brother and evaluating all the different ideas for delaying his ALS, I built a very rigorous pipeline tool and I managed all the choices, and that included supplements and stem cell transplants and different drugs, very much like the longevity movement today. But my timeline was very acute. It's such a desperate disease, or motor neuron disease, or ALS. And I realized that I did not have a tool to understand whether something I was doing would working in my brother. And that really was a hampering, even though we did multiple clinical trials for him and the stem cell transplant, hampering our ability to be aware whether we were making progress. And that really inspired this idea of proxy markers, or this idea that you could have something that you could know before the actual event happened as to whether that event was going to be significant or change your life. And Alden was founded because I believe that through some of my own work and work of others in the field, that we had reached a time in biology where we had the technology in machine learning, or if you want to call it artificial intelligence, and biological measurement to truly understand human complex disease at a new level. And we could leverage that to build biomarkers for the things that we want: fluid intelligence, vitality, strength, the things that we want. Not just longevity is less of the issue, but like, how do I be my most vital, intentional, powerful self in the world? Mm-hmm. And I realized that none of the data that I was dealing with from clinical labs, or the data from the papers or the information that's coming out that's exploding about understanding aging and health, really could be brought back to me in context to give me a tool in me as an individual to measure something. And so Alden Scientific was designed to provide those markers for the first time in a rich way in an individual.
Okay, so that's the why. So tell me about the what. What is Alden Scientific?
Yeah, so Alden Scientific is it's a biomarker clinical decision support tool that essentially enables you to draw blood either in a research study or as an individual in partnership with your physician, mm-hmm. and use state-of-the-art research-grade assays under an ethics framework, and then evaluate the differences in those assays, just like we evaluate distance using GPS, to the things that you want and the things that you don't want. So, and then to use those distance metrics in certain ways to provide a position of where you are in the journey of life, and to give you guidance and advice vis-a-vis the biology you might want to change, or ultimately, though not mm-hmm. quite yet, the interventions you would want to take to move you to that place, and to do so using all the known information, not just prior opinions from others.
So that's interesting. So there are two markets for your products, listening to what you just said. That one of those is obviously the clinical community, and another is the research. So yeah. Are you deployed in both at the moment, or are you experimenting with one first, then moving on to the next?
We are deployed in both. As an MIT grad, I'm kind of proud of the fact that MIT is our first customer, and for them, they are running a study to look at people who have post-infection syndromes, so chronic Lyme or chronic COVID, sometimes unknown infection syndromes that look like that, or active infections. And so there's a clinical research study going on, and we are processing much of the blood to provide an interpretation of the differences between those groups and what it means.
Mm-hmm.
But we do so in the context, not just in that one study, but by using very large databases like the UK Biobank, which has a large cohort of 50,000 people that have been measured comprehensively using a similar technology, or our own data that has several thousand samples that were very specifically measured around the symptoms of inflammatory disease. And that information allows us to tell MIT their particular finding. It's a Professor Mickey Tao and the Francisco Maestro. But the beauty of what we're doing is that we have members, which are our early investors and adopters, and then some scientists that many of you would know. That those early adopters are working with us, and they are also getting the same processing and the same samples done, and they are getting individual data back. And what I love about the combination is that there's a particular theory for how antibodies relate to pathogens that this Professor Tao has that I think is really thoughtful. And if true, and if we show it, our members will have that information immediately. Mm-hmm. So not waiting for the publication, not waiting for the 15 years of dissemination, but immediately. And the same is true of some research we're looking at in space medicine. The same is true of some research we're just starting in PD-L1 inhibition in oncology to prevent remission. These kinds of things, I want a translation time of 0. Mm-hmm. So that when the discovery is made, that I can send me and my family and our friends and our members a graph that shows where they are in that discovery the day it comes out.
Okay, so just referring to members then, so you've got the scientific community maybe using you as a research platform, you've got the clinical community, which we'll come to in a minute. We've also got members, right? So yes. How does that work?
Well, I mean, one of the beautiful things about the longevity movement is a lot of people are self-experimenting. And these are resourced and thoughtful people that are trying ideas. I do some of the sort of high evidence, low risk supplementation or dietary changes, behavioral changes like fasting. And then there are more aggressive ones like rapamycin, or even more aggressive things like some people doing some crazy gene therapies, or doing broad stem cell transplants, truly off label in ways that they believe will change their health outcomes. And my belief is that we should leverage that. Having gone through a cycle of this in ALS, where I watched people try things and then the information was lost to the world, the failures or even the potential things that worked. If our platform was fully deployed at scale for people that were trying things, before or after you did anything, could be a fast, could be starting rapamycin, it could be a stem cell transplant, it could be some other new invention, or some of the more effective supplements that I think are interesting. And the before and after allows us, because of the way we do our mathematics with the right time dimensionality of biological features, to say, well, in the short term it does this, and in the medium term it does this, and in the long term it looks like it will do this. And that's the same information that we want from a drug development program, the same information we want for discovery. So I think what's amazing is that with machine learning and the right biological feature measurement at scale, we can converge discovery, development, and the best of care. Okay. And that's what I think we're doing.
That's very interesting. So obviously, some people are experimenting with quite novel therapeutics offshore. Are you saying that if somebody was using your system to manage that in a clinical environment, using the third pillar of those three types that you've got, that they would be not only drawing on other members of a similar cohort, but they would be backing into, let's say, an evidence base from UK Biobank, which is also supporting the pathways from novel therapeutics that have been in the marketplace for perhaps 20, 30 years.
Yeah, and you're hitting at, that's exactly what it's for. And in the ideal, we'd be able to do an N of 1 experiment. Like the measurements would be so good that in one person, you could measure the change in all the dimensions that mattered in a way that told you whether that thing was working in that person. And I don't know that we're there yet, and we're not quite there yet, but we're working on it. But in the journey of what we're trying to do, this is my prior work. I mean, at Patients Like Me, which I love in the name, Patients Like Me, the idea was to actually embrace the heterogeneity of the human variation and understand how to adapt patients. And what our business was, in large part, was helping pharma companies design better trials, balancing the differences in patients rather than saying, I'm going to falsely blind them in this double-blind ideal standard, which is wonderful as an ideal. But the real world is much messier than that. And longevity is not going to collapse to a double-blind study because no one's going to wait to die. It's just not going to happen. So we need to move to this proximity to causality, this understanding of human variation. And the more we can account for that and vary that, and I have several publications on this in work and trial adaptions and in using control groups in different trials to get better data. But with the data we have now with this sort of machine learned biology, it is so precise and so capable of managing nuance that if we get the question right, I think we can really get down to the place where in an individual or a very small number of people, 5, 10 people, begin to see whether something actually works. And why that is so important to me is I don't see any reason why we can't functionally extend human life by 20, 30 years. And this is just like the genetics says we can. The environmental variation says we can, but we don't have as any decent measures of whether we're succeeding at doing that right now.
Mm-hmm. Yeah.
And so if we get those measures, it will happen. And I think that, to me, I want out in the world with as many people as possible because I don't want to have to try everything myself. I want to learn from everyone else. And so it's a sort of a confidential social network for people that are committed to understanding and advancing their own health. Now, at the moment, it's very high cost because these assays are very expensive. But we have a very clear path to lowering this price point over time to a place where it can be broadly acceptable. But right now, this is like private jets for personal discovery to understand how to not die.
Mm-hmm. So let's talk about why you're breaking ground now, right? So you're coming out of this period of time where your website's pretty opaque at the moment. Yeah. You've got a combination of 3 groups of people. Some of them are private individuals. Some of them are leading clinicians and researchers. So where are you now, and what's the next stage of your evolution?
Yeah. So the important thing that shifted this year was that for the first time, a large public database was released that changed our ability to create a digital twin. And now people say they're doing that with health records, and they say they're doing that with clinical labs and all that stuff. And I've done all that work over time in multiple publications in different ways about that. But none of those were very accurate. And you didn't feel like you knew the individual. But when you use proteomics at scale, analyzed correctly with the right timeframes and understandings and what biological variables belong in and out, we can compress a human being down to a very small number of numbers that are orthogonal independent and tell us why we're each different, which is so critical. We're different. So that data we knew was coming because we'd done a similar experiment ourselves. We knew the data that we have now again, that a few years earlier. And so we prepared for that, built all of our platforms, the cloud computing, the analytics engine, some of the tools we use for displaying the information. And when that became released, we started working with it. And the reason we're operational now is that we're so confident in the quality of our models. So, for instance, in cardiovascular disease, we are twice as good as all of the risk estimators that are in the world, like the American College of Cardiology. For Alzheimer's, we have an initial model that is 50-fold stratification for your risk of Alzheimer's, which means if we think you're not going to get it, you're not going to get it in the next 15 years, really. Almost
so insurers are next on the list.
Yeah, I know who my customer is. My customer is the person the world needs to give tools to individuals to choose well. Mm-hmm. We don't have a great marketplace for diagnostics in healthcare that does that. We don't have the insurance marketplace for health insurance is broken. The life insurance marketplace is fine. You want to bet on life, you can do that. We can help you with that, maybe. What I'm more interested in is with individuals, giving them the tools to show them how they can move closer to the center of that warm river, the journey of life and the sun and the joy, and away from the Alzheimer's and the strokes and the heart disease, and navigate that by their own choices. And, a good example, my brother, who's a member and another venture capital investor, he said to me, and he'll forgive me for this, but like he says, okay, I have GERD, but I'm not going to stop drinking, and I don't want to change my diet. So what can you do? And I think to me that mathematical question, which we can work on, and we are, and are finding sort of biology that is sort of independent of those things, that he may be able to adjust to reduce his GERD without that. I love that kind of request. Mm-hmm. So this is not some like, how do you behave well? How do you do this? This is like, because I love to have a Manhattan, and I enjoy my life. My purpose is joy. I just want to know what the things that I can do in the context of joy that will help me be more vital versus less vital. And that's the sort of level of skill. And I'll give you one other fun experiment just to show it off a little bit. You probably can just see it. But there's like 5 dots on my arm here. 6 dots.
Is that
I barely see them. But on Wednesday last week, to demonstrate why our current tools are so bad. And these are in use by essentially every major longevity company in the world, right? I order the same 100 bank of labs that everyone else does all the time. So on Wednesday, I woke up at 6 a.m. and my phlebotomist was in my kitchen, and I rolled downstairs, and without even stopping at the bathroom, I had my blood drawn. Then I had 3 cups of coffee and a banana. And at 9:30, I had my blood drawn. And then I climbed up to the office, which was, our office is 12 flights of stairs, and I had my blood drawn. And then I had a beautiful caprese sandwich from this really neat place called Brothers in Cambridge, and then I had my blood drawn. And then I had a Gatorade, and I had my blood drawn. And then for dinner, it was a very nice dinner. We have a place called Row 34 across from the office. 2 martinis, 12 oysters, and a lobster roll. You have to do this in Boston because the lobster rolls are best here. And then I had my blood drawn. And during the day, according to the current criteria for how you would do medical care, I was at one point essentially the healthiest 57-year-old you could measure. At one point, I was hyperlipidemic. And at one point, and I probably would be put in a statin. And at one point, I was clearly hypertensive and would have been put in a statin. So these are the tools that we use for care today that in one day I managed to change in my body. And the whole point of this experiment is to show that our tools are stable to that they can manage that variance. That to me, it is the total of your behavior that produces your outcome. It is the total of your interventions. And that's just one of the kinds of things that we're doing. And each of those blood draws is going to cost $6,000. So we're not talking about small money here.
Yeah. So how do people get involved? I mean, I guess there's thinking about the community that we talked about. Obviously, there's the scientific research community, there's the clinical community, there's the user community, maybe even the investor community that want to participate in, what you're doing.
Yeah. So getting involved right now. So we're raising money. We're finishing a safe note that's been funding the company. It's got about 10 million total in it, and it's the very end of it. And we have a little bit left in there. And then we're going to have a round either with one of the 2 of the strategics we're talking to, or several of the VCs that we've been talking to that are in probably big due diligence. So we'd welcome other parties. I am very interested in people that are really interested in understanding the objective. The purpose here is to understand ourselves, use it to leverage it for our own health. That's the first, second, third purpose. And I want funders that are aligned with that objective. I think this is the best way to build the most valuable business possible, but we're going to do it that way. But the way to get involved right now, it is essentially people that are either adding value because they're scientifically involved. So I have a partner that's a company that we work with in the microbiome space. We have several other bioassessment companies. And so some of those people are enrolling so they can understand their own technology better. So people that do proteomics or people that do microbiome research, glycomics, whatever area of domain we're looking at, epigenetics as well. The second thing is a group of people that have been early investors. And the early supporters have to have an additional requirement. So this is a very, very powerful tool. When you see what it can tell us about the future, but preliminarily, first generation models, and they're good, and we're not, these, I want it in the hands of people that understand 3 things, which is basically, do you understand what a type 1 error is? Do you understand what a type 2 error is, right? False positives and false negatives. Do you understand the consequences of numbers on those two concepts. And then the third is there's an Italian statistician named Bonferroni. They developed a correction for t-tests. T-tests are the common scientific way of saying something significant. So every paper you and I read all the time about showing, oh, this has shown significance in extending life, and this has shown significance in say, like, that's always a t-test or some other statistical methodology that does that. And Bonferroni developed a method for correcting it. And to me, if I ask you 20 questions, and in each one of them is a 1 in 20 chance of being correct, one of those will be positive, and it's not, it's a lie, it's a false, it's spurious. And so if you don't understand that at this point in the discovery, when we're looking at 50,000 or hundreds of thousands of variables in combination, that some of them will be spurious and therefore be reversible. Mm-hmm. And that affects the type 1 and the type 2 errors we're calling, then you can't get in. And that's a very complex ask, but like, I want people that want to know themselves, that want to try things to make themselves healthier, that want to use information in a safe and effective way, are going to be responsible with their claims. And also understand we're going to make mistakes.
Okay. And how do they get in contact with you?
Well, you can reach out. You can reach out directly to the login on the website, where you can put your name in. We'll get back to you. You can find me on LinkedIn. Through someone on the connections. I'm sure they can reach out through you. And the cycle is going to be, look, we have 50 members now. We're going to probably go to 100 by the end of the summer, maybe 200 by the end of the year. And I think we only really have room for up to, say, maybe 1,000 next year, even with all the scaling we're doing. And these blood tests, we measure everything that can be measured. Like, we're talking, epigenetics, like everybody else does, RNA, DNA, whole genome. Obviously, those are obvious. But these, 1 or 2 of the highest dimension proteomic assays in the world that are not available, you can't buy these anywhere, or advanced glycomics, that's every measurement. We culture the microbiome. We are looking at, cytoph and different cell counts in ways that are just never done in general. And so this is the difference between GPS and a sextant and a compass. It's that different. I just don't have a GPS level map yet.
So, Jamie, just to help ensure that you don't get a tsunami of people that perhaps can't come up to the requirements that you need in terms of the finances, I mean, what are the sorts of clients that you're looking for?
Well, I mean, right now it's sort of the alpha is this sort of 4-year self-discovery program. We're looking at 3 to 6 samples a year. You need to bring your own doctor that we approve of. We have to interview the doctor. They have to pass similar kinds of questions, and we have to like them. Like, we're looking for good energy. We also are biasing to people that have a disease interest. So, the MIT project was supported by someone that donated, several $1 million to MIT. We're only part of the research project. Things that add value to the common corpus of information that we could do. So if you're interested in studying your disease in some way and you want to work with us to make it more effective, that there is a little bit of a look. I mean, it's very expensive. We're charging. We're planning on at the moment 200,000, or it might even be more, for participation. But I think we're still actually, given that we don't have a lot of capacity to do the kinds of individual analysis we want, we're really interested in karma. Like, this is very, this is stuff where you show us how you want to make the world better, or you believe in that, and that's something that we can invest in you as well.
Well, Jamie, it sounds fascinating, and I think I understand it now, which is much better than when I was at the beginning of the conversation. So thanks for joining us today, and obviously, best of luck with everything. It sounds fascinating.
Yeah, it's such a pleasure. I have to say, this decade is going to be so unbelievably exciting in what we can do to understand and change our lives. And it's so fun to be in the center of understanding it. I've never had a more enjoyable experience in my life, and I appreciate the interview today.
Great. Well, this is your 4th or 5th one in, right? So this is going to be the big one.
Last one, big one. Well, yeah, exactly.
The last big one. Great. Jamie, thanks for joining us.
Bye.